In a previous lecture, we equated the word potential with voltage. We said that basically potential means electricity in the human body. And the term voltage has a definition that means a separation of charges, which is what a membrane does. It separates the ions on the inside from the ions on the outside. Every cell in your body has electricity. In fact, every cell in your body has what is called a resting potential. And every cell in your body has a certain value for that. And we can put a number on it in terms of millivolts, but every cell has this resting potential. Since we're starting to talk about nervous tissue can have graded potentials. And these are like a little jolt or change in voltage, but it dies out like a match. You light the match and then it dies out. You can also have what's called an action potential. And that's like TNT, where you light the fuse and the fuse goes all the way to the end. Let's take a look at this term here for an action potential. It's a stimulus, so that means we're changing the voltage. It says that it's propagated, which means that it moves. It also mentions two parts of the neuron that are really important for us to remember right now. This area right here with the black dotted lines around it is the axon hillock. It's an area of resistance, and that's why I like the word hillock. It makes you think of a hill, something that you have to like climb up to get down over again. And then this area here is the axon itself. So neurons will receive stimuli up here in the cell body, and that stimuli will be propagated or moved over this area of resistance and down the axon to another cell. We've previously talked about this other cell being a muscle, but now here we're going to consider it being another neuron. We're curious first about action potentials, and then we'll talk about graded potentials. So an action potential, I mean, it depends on what kind of book you learn from or what class you take. Some classes will call it a thought. It is. Some classes will call it an impulse or a signal. I feel like those words are really vague. But no matter what you call it, we're talking about moving electricity over a long distance. And that's what the axon is, right? The ends of a neural cell, the teleodendria, are a long distance from the cell body. Only neurons and muscles can have action potentials. All of your cells have resting potentials, but only neurons and muscles can have action potentials. An action potential, it technically is like a washing machine, a series of predictable changes. So the washing machine goes from the pre-wash to the wash to the rinse to the spin, and it goes in that sequence. Here, the membrane changes voltage. It goes from -70 to -30 to +30. It just changes in its voltage. Why does a membrane change in voltage? Well, you might remember voltage-gated channels. I equated them to like the key card that you get at hotels nowadays, right? You put the key card in and you break the magnetic strip and you're able to walk into the door. So there are some membrane channels that change with voltage. We previously talked about membrane channels like this one over here that opens with acetylcholine. However, these two channels over here, these two channels open from a change in voltage. Notice how there's no acetylcholine anywhere. The first thing that you need to know about an action potential is that the series of predictable changes, they all happen or they don't. There's no half action potential. So when we hit a certain threshold, it's kind of like all of the steps of an action potential will proceed from start to finish. And we will define those steps. What I'm trying to say to you here, though, is like, why do I have a pregnancy indicator here? So you can't be half pregnant. right? There's either a plus or a minus. I mean, you may have gotten one of those tests where you're like, I can't tell if it's a plus or a minus, but it's either a plus or a minus. There's no half there. There's no start then stop. An action potential either happens or it doesn't. Before we can even look at a graph having to do with an action potential, we have to kind of understand what the graph is. Let's think of a neuron with its axon. And what this graph is showing us is just one point on that neuron's axon. Just a place where there's a couple sodium and a couple potassium channels. Pick a point. Do you want it to be this one or do you want it to be this one? But what we're saying is that the changes we describe in this graph will happen successively at every point along a neuron. It's kind of like the wave in a stadium. You have to think of like the wave starting up here in the cell body and going all the right. Everybody stands up. So let's understand this graph. Let's put some labels on this graph. Down here, this is time. And it's not seconds. It's actually milliseconds. So that's really short. Over here, this is voltage. You can label it as voltage. Another word that you could use is potential. And that would be more correct because we're talking about voltage in the human body. Are these volts? No, they're millivolts. Okay, all right. Let's define some things that are already on the graph, like this dotted line and maybe some other stuff that's noted here. We have -70 down here. And -70 is the resting potential for many of the cells in your body. So that's the voltage at which they chill. That's the voltage at which they chill. This point here, -55, this point is called threshold. This is the point at which we have an action potential and there is no turning back. Okay, students usually get these two mixed up. For some reason, they think that the dotted line is resting potential and then they get confused. So don't forget, resting potential is always more negative than the threshold, always. So what we're seeing here is that as time proceeds, there will be changes in the voltage at one place in a cell membrane. It seems here that we have some changes in the membrane that make it more positive. Yes, and those changes in a membrane are called depolarization. That word, depolarization, just means to make more positive. Over here, we have something else, and this is called repolarization. And this term repolarization just means to make more negative. We also have something else that occurs down here, and we're just going to label it nebulously right now, hyperpolarization. We'll talk about that one, but let's start by considering that depolarization and repolarization are opposites of each other. Okay, I want to show you that there is a limit. We don't have a name for this limit up here, but there is like a limit, a topping out, where the action potential, where the axon cannot become more positive. So there's a limit to this. It's not completely positive. Ooh, it's not completely positive feedback, which you see on this side of the graph. An action potential also incorporates negative feedback, bringing it back to homeostasis, which is your resting potential. Okay, this graph has a lot of stuff on it. Let's do one thing first, and let's highlight all the terms that we already know. We know time. We know membrane potential, millivolts. We know resting potential. Let's look at it here. It's -70. Okay. We know the threshold, and it's a little bit different on this graph. It's -60. The previous graph was -55. There are some other terms that we actually talked about here. There's depolarization. Look, that's on the uphill. There's repolarization, and that's on the downhill. There's some colors here that say absolute refractory, relative refractory. We'll talk about that. I want to show you, I like the steps in your book. Let's just look at them and what they mean on a graph, and then we'll look at them more specifically. But there's a couple of things you want to always look at. You want to look at how the charge is changing. Are we becoming more positive or negative? And you want to look at what kind of channels are open. Are they sodium or potassium? Let's draw ourselves a little diagram down here in the bottom to remind ourselves of something that we've learned all throughout this class pretty much. This is a cell. Potassium is common on the inside of the cell, and therefore potassium likes to move out. Sodium is common on the outside of the cell, and therefore sodium likes to move in. So if I open channels, these ions will just move, right? And when sodium moves in, the cell becomes more positive. When potassium moves out, the cell becomes more negative. Let's go over that again. The first thing that happens in an action potential is actually a graded potential. And your book doesn't call it that here. It calls it a graded depolarization. We can call that a graded potential, right? So a graded potential happens, and that brings us closer to threshold. We don't pass threshold, but we get closer to threshold. Once we pass threshold, we're going to take off with all of steps 2, 3, and 4, and there's no going back. Do you want me to, I want to abbreviate step 2 for you here for a moment. Your book says voltage-gated sodium channels. Step 2 is depolarization. That's what step 2 is here for you. It's just depolarization. Depolarization is when sodium channels open. Sodium rushes in. The cell becomes more positive. You want to know what step 3 is? Step 3 is the changeover from depolarization to repolarization. So no more sodium, but we're going to start to open potassium channels. And therefore potassium is going to move out, and the cell's going to become negative. Now what happens is that the cell overshoots its resting potential, and that's an issue. We have to get back to -70, and as you can see down here, it's kind of like we overshot it, right? So what the cell does over here is that it will open sodium channels and close potassium channels to return it back to resting potential. So it'll make a decision. The other thing that is active over here is the NAK pump. This pump is desperately trying to return ions to their rightful place. Let's put that into words here. Let's put it into our words. An action potential has 4 steps. The first step is a graded potential. And that graded potential brings us closer to threshold. Here's resting potential. Here's threshold. Okay, we don't pass threshold, but we get closer to it. In order to do this, we have to open sodium channels. And that's what you see here in this orange line. It's showing you that sodium channels are opening. The next step is that we pass threshold and we get like a full depolarization. And that's this area right here, and it's the same thing. Now we don't have some potassium channels open. Now we are in the process of opening all of the potassium channels. The peak of this action potential right here is when all sodium channels are open. This is when the cell is saying, I can't possibly open any more sodium channels. I can't possibly become more positive, even if you wanted me to. The third step is repolarization, and that's what's happening here on the downslope. And as you can see on the downslope here, we have potassium channels opening. And that's what's here in this green line. You're seeing potassium channels open. And then, because you know that we overshoot our resting potential, we tried to return the cell to its resting potential, but we overshot it. And then in hyperpolarization, we are closing potassium and moderating sodium. And don't forget, in hyperpolarization, the NAK pump is also desperately trying to return ions to their rightful place. These are the steps. Just memorize them. Just remember that when you say something like depolarization, you have to think about what channels open, what ions are moving, and in what direction. Oh, look at that, NAK pump. The next thing that we have to consider about an action potential, it's something called a refractory period. And I like this graph in your book because it shows you that the absolute refractory period is like shaded in red here. And the relative refractory period is kind of shaded in gray, but it's like, I don't know, a little purplish. But refractory periods are periods when the neuron can respond to another action potential. Think about that again. An action potential proceeds, but then the neuron can respond to another one. Let's see, though, if there are some rules to that. The first refractory period is the absolute, and that's when, no, you can't. You absolutely can't respond to another neuron. You have started steps 1 and 2 of the action potential, and there is no going back. You can't respond to another stimulus. And that has to do with the fact that at the end of the absolute refractory period, at number 3 right here, all of the sodium channels are open. You couldn't open any more if you wanted to. This is like when I say to you, oh, maybe you could study more, and then you say to me, Amy, but I am studying as much as I possibly could. Like there's no more study that I could possibly do, right? You're at the tip, you're at the peak. You can't do any more. However, when sodium channels start to close, which is what happens in this area here, they start to close. We can respond to another action potential, but it has to be stronger than the first one. So, for example, you go to lift a bag off the floor and you contract your muscles a little bit, but your brain realizes that the bag is heavier than you thought. So there are a few seconds, about 0.5 milliseconds, that you can't make your muscles contract anymore. But then after that, they can. So that's the relative refractory period. The difference between these two is the state of sodium channels. In the absolute, there are no more I can use. I can't recruit anymore. In the relative, the sodium channels start to close, and therefore I can get another action potential by reopening them. Okay, all right, so we've talked about the steps of an action potential. Let's take a step back and look at a neuron, because what we were looking at in the previous 2 graphs was like one little itty bitty place on a neuron. So that graph happens in each of these places. And remember what I said, as an action potential moves down an axon, each place goes through the rise and fall into voltage. It is just like people in a sports stadium doing the wave. Each person manifests that graph at that point in the axon. They rise and then they fall. So propagating also means conducting. That's another word that your book uses, conduction. And these 2 words are 2 words that are used to imply that an action potential is moving down an axon, specifically the axon. In white matter in your body, you have myelin. And therefore, when the action potential moves down the myelinated axon, it goes like this. Ding, 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 jump, ding, 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 jump, ding, 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 jump, ding, 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 jump, 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 until the end. I'm sorry, I couldn't help myself. This is really fast. This is what's happening right now in your sciatic nerve if you wiggle your pinky toe. You have a cell body in your brain, and then the nerve comes down your spinal cord, out your leg, and into the muscle of your big toe. That's pretty fast conduction that you can say, clench, contract, and your toe contracts. This is what is called saltatory conduction. Saltatory or saltaire means to jump. That's what the word means, to jump. And that's what your action potential is doing. Your action potential is jumping between the nodes of Ranvier. And that's because only in those nodes of Ranvier, those are the only areas where those ion channels aren't smothered. The myelin is smothering the ion channel, saying, nope, jump over to the next. No, jump over to the next. The axon on the bottom is unmyelinated. There's no myelin on it, and that's what we call gray matter. This has very slow conduction. This is where the action potential has to go through every single ion channel on the axon. This is like going in the supermarket and stepping on every single tile on the floor. White matter myelinated conduction is like jumping on only the dark tiles, so you jump. So gray matter has very slow conduction, and this is what is called continuous. Continuous because we're not jumping from node of Ranvier to node of Ranvier. This is what we can relate to multiple sclerosis. In diseases like multiple sclerosis, the myelin sheath here starts to get broken down. And in some places, the myelin sheath can be degraded completely. So it's like maybe this sheath is gone. Maybe this sheath is gone. And what happens is that in MS, an axon that was myelinated turns to unmyelinated, and saltatory conduction turns to continuous. People who have MS like demand their limbs to do things and nothing happens. That's because the action potential does one of two things. It either takes too long to get there because it's in continuous conduction, or it never gets there, which is what happens in the later stages of MS. How fast? How fast does that really mean, right? How fast? We understand that a myelinated axon has faster conduction than a continuous axon. So like a myelinated neuron might be one that descends down your spinal cord, whereas an unmyelinated neuron might be completely in your brain. That's your brain. Okay, so it depends. How thick is the axon? How myelinated is the axon? So we classify nerve fibers in your body. Let's think of your brachial nerve in your arm compared to your sciatic nerve in your leg. We classify those nerve fibers by these two aspects. The sciatic nerve has a big diameter and lots of myelination. Your brachial nerve doesn't have much of a diameter, but it is myelinated. Neurons in your brain have a very, very thin diameter and are not myelinated. So we classify these into A, B, and C, and you can see the delineations here. So your sciatic nerve would be a class A nerve fiber. And your, you know what? Like your trigeminal. Now, let's just say that something in your brain would be a type C nerve fiber. Why do we care? Because different drugs act on different nerve fibers. Botox likes to act on the C's, doesn't like to act on the A's. That's why you don't get Botox into your sciatic nerve, but you do get it to your trigeminal, right? Different drugs that work for Parkinson's and MS have different effects in neurons in the brain, which might be C, than they do on neurons in the body, which might be A. So to treat your brain and your body, you may need two different drugs. And again, that's just the difference in the environment in your peripheral nervous system, outside in your limbs, in the trunk of your body, and in your central nervous system, your brain and your spinal cord.